All right. Good afternoon, everyone. Thanks for coming to our EEB departmental seminar series. I'm Colin Kramer. I'm organizing the series this year. I'm delighted to introduce today's speaker, Dr. Allison Moreno. She's an LA native who does awesome research integrating ecology, oceanography, organismal biology. She thinks of herself as a marine biogeochemist and a phytoplankton physiologist, which of course are topics near and dear to my heart. But she followed an interesting path into this career, starting off with an obsession with sharks, mako sharks, and great white sharks. If you're talking with her later, ask her just about anything about sharks, maybe.
Yeah, I mean, just about the ecology.
She studied at East LA College and CSU Monterey Bay before doing a PhD master's at UC Irvine with Adam Martini. And then coming to us here at UCLA as a chancellor's postdoctoral fellow in the Atmospheric and Oceanic Sciences Department. And we're just really excited to hear about your work today. Thank you for coming.
Awesome. Thank you. Thank you, Colin. That was a great intro. It is true. I love sharks. Sharks are my life. And the reason that I started doing phytoplankton in the first place. So a lot of the research that I'm going to be talking about today was actually taken, a lot of the samples at least, were taken while at UC Irvine. And so because of that, I want to start by going ahead and thanking them for a lot of the funding that went into this. However, a lot of the analysis and some of the maps you're going to see today were done while being here at UCLA. All of my funding sources, thank you so much for being here and giving me a good amount of money, because some of this work did take a minute to do. We are all fundamentally doing something to survive, not just listening to my talk, which that's a good thing. We are also breathing. And if we're not someone who thinks about breathing on a daily basis, because that's not your research, what you're doing is you're intaking oxygen, you're releasing CO2, fundamentals, but what you're really doing is producing energy for yourself. Now, we always think about breathe in, breathe out, we're stressed out, our lungs are expanding and contracting. In addition to that, though, we need to consider how that can be seen from other animal perspectives. So what we do have are, of course, aerobic respiration that is going to be occurring within mitochondria and into the cytoplasm within your cells. If we don't allow this production of ATP and energy, then our cells aren't going to function as well. So oxygen in and of itself is really important in order to make this process occur. Now, we may not think about the different ways that we can have breathing occurring in other organisms because we don't really care, right? I, as a phytoplankton person, I don't really care how a chicken breathes, but they're really cool. And so we might want to consider how they're breathing and how that's extremely different than looking at a sponge or a fish, right? There's a different type, lung versus gas exchange. Either way, they're still respiring and taking in oxygen in order to produce energy. Now, of course, this isn't just common in animals. Plants of all sizes are doing that. If you've been anywhere on campus, which I know all of you have, because you had to get here, you've seen tons of different sizes of plants. All of them are going to respire at night, and they're able to do this exchange of oxygen to CO2. We like to think about their photosynthesizing properties and how they're producing oxygen for us. Sometimes we forget that they're also doing respiration. They're important, but so are the microscopic plants in our environments, such as phytoplankton. I personally love phytoplankton. They are the fundamental basis for the food web within the oceans. They also are producing photosynthesis as well as respiration at night. Bacteria and zooplankton are also going to consume them. And so we can see that if there's changes in phytoplankton, that's going to have very large effects potentially on some of our larger organisms, such as sharks. Phytoplankton also are one of the most prominent producers of oxygen. So they can produce anywhere between 50 to 70% of the oxygen we're currently breathing. So the next time you take a breath, think a phytoplankton if you are inclined to do so. Although they are doing this kind of from a byproduct point of view. So in order for them to have sugars and to survive, they need to produce oxygen, okay? They use CO2, water, they use energy from the sun as well as different nutrients in order to produce chlorophyll, sugars, and that byproduct of oxygen, which we of course appreciate. as phytoplankton continue to do this, producing all of this oxygen, you may think, well, what is it utilized for? Obviously, fish and other marine organisms, as well as bacteria. And bacteria have a very fundamental use. They like to consume dead phytoplankton. So as phytoplankton are at the surface, living their best life, at some point they start to die off and move through the water column. As they go deeper into the water column, they're then consumed by bacteria. And when they do that, bacteria are using oxygen in order to consume them and create areas of low oxygen. Now, these are oxygen minimum zones or oxygen deficient zones, depending on which realm of research you're in, basically low oxygen spaces. And these are found globally, and this is very much a natural process. And we call this process deoxygenation. However, in and of itself, this process is going to be affected by anthropogenic inputs. And so I said this is happening everywhere. So where exactly might we see this? In blue, we have areas in the open ocean that are low oxygen. And in red, we have coastal spaces that are exhibiting low oxygen. Now, this is globally, we're seeing that there is quite a lot of areas that have low oxygen. However, one thing I want to make very clear is it's not uniform. So in the Pacific Ocean, we have a lot less oxygen cumulatively than we do in the Atlantic Ocean. In the Atlantic, we have quite a bit more. So we can see that both from the visual point of view. We also can look over on the other side here and see that we have the dissolved oxygen on our x-axis and going down deep in through the water column. The differences between in blue, the Pacific Ocean, and the Atlantic in red. So it's very clear that we see that there's oxygen decreasing through the water column, that we have a certain band, right? So around the anywhere between about 150 meters to about 1,000 meters deep, you can find low oxygen spaces. This is, in and of itself, one of the three major threats to marine life. It may seem extremely obvious if we just consider us, what happens when we go into low oxygen spaces, but this is really important when we consider all of marine life and what implications they might have. So it is exacerbated by ocean warming from both a stratification point of view, but also from the point of view of solubility. So temperature has two different effects. We have the solubility factor and we have a stratification physical factor. Additionally, a lot of the spaces that we're looking at along the coastline, especially down here, we may be thinking if anyone is interested in rivers, Mississippi River, we do have a lot of eutrophication as well as agricultural runoff that can cause these low oxygen spaces because they're influencing phytoplankton in the space. So I said oxygen is important to marine life, and I'm hitting you guys over the head with that. Like I really, really am, because we need to consider that with low oxygen now and expected to continue to decline into the future, we're going to see large biological productivity and diversity decreases. So large reductions in that. And here, some people like to tell me, you just stole a picture of a coral reef bleaching. In fact, the research that was done in this space was not only seeing warming affecting coral bleaching, it was also affecting the keystone species that were there. They all migrated out of the space and coral bleaching was occurring at the same time. So you can have these double effects impacting large spaces. We've also seen that there are major changes in the animal behavior, so migrational patterns of large species like tuna. We've also seen really cool stuff with sharks, shark attacks occurring in regions that had never seen shark attacks before, simply because they were essentially stuck in the coastline by these low oxygen waters, creating a barrier. Both of these things, having this reduction in biological productivity as well as changes in animal behavior, are going to have strong implications on fisheries, not just from the point of view of we're going to fish and there are no fish, but also the time and energy spent going to do these fishing. Areas that completely are dependent on fish will probably not have fish in and around their coastlines. And so it's something that we need to consider from a big point of view, not just thinking about oxygen in and of itself, but the implications of lower oxygen. Okay. Cool. Great. What does oxygen look like though in our oceans? So here we're looking at 200 meters deep. This is the area estimated to be one of the best parts within the ocean to look for low oxygen spaces. And we have on this end data. So we have the cooler colors in blue, being lower oxygen, and then the red colors being higher oxygen. Typically, you are going to find at your high latitudes, both the north and south, that they're going to have the higher amounts of oxygen because, again, solubility properties. And then same thing with the equator. Warmer waters means less oxygen, typically. Now, you can kind of see, I feel like it's very obvious on my computer and not so much on the screen, but modeled data shows a lot less oxygen in the equatorial regions. Why? Because they're using a value to convert essentially from oxygen and carbon, converting those two factors with a static value. Now, this value we're going to call the respiration quotient, and I'm going to get into details about that. However, because we're doing this, we are underestimating how much oxygen is in our oceans, but it could be that we're also overestimating in certain spaces. So really the region is going to change and it's going to matter. Okay, so to follow through with my talk today, we're going to start going into the nitty-gritties of what is the respiration quotient. We'll talk about some of the very first direct quantifications in the Pacific Ocean. From there, we'll be able to generate hypotheses as to how the variation that's occurring in the respiration quotient is acting. We'll then test those hypotheses in the Atlantic Ocean, and then we'll get a sneak peek of some future potential implications when we really think about oxygen as a whole. So what is the respiration quotient? It is the amount of oxygen needed to fully oxidize all organic carbon. So I'm going to do this chemically, very simply. We have a 6-carbon sugar, so glucose, plus oxygen, then yields CO2 and water. We simply get the coefficient in front of the oxygen and divide it by the amount of particulate organic carbon. Therefore, we get a value of 1 in this specific scenario. However, we know from understanding of carbon and the oxidative states of carbon that the hydrogen ions actually matter to the specific particulate organic carbon. And so because of that, we actually have an extra little factor that comes in that accounts for oxidation state. But ultimately, even though we can get really complicated by adding in nitrogens, phosphorus, you can really go as far as you want, silicas and whatnot, you're still going to get the coefficient in front of the oxygen utilized, divided by that particular organic carbon. Now, a slight aside, because I know we are not dealing with a room full of oceanographers, Alfred Redfield is an American oceanographer who quantified a similarity between surface plankton, elemental stoichiometry, so carbon, nitrogen, phosphorus, oxygen was later estimated, with that of deep ocean dissolved nutrients. And what he hypothesized is that this is because of phytoplankton. And if that's true, which it in a lot of ways is, he found that the stoichiometric ratio between carbon, nitrogen, and phosphorus was 106 to 16 to 1. And we can essentially create what Alfred Redfield's Redfield ratio is and allow for respiration to occur. Okay, his value essentially for the respiration quotient would be 1 as well. He makes a very strong assumption, and in this room, it actually is impacting and great. He assumes that all carbon is coming from carbohydrates only. And we all know that that is not true. A lot of people don't realize that carbon is in a lot of things. And we have cells that are very dynamic and very different. Here I have a picture of a cyanobacteria. Here is a picture of a diatom and just a very fun cartoon of the different organelles that are found within cells. Every single one of these organelles contains carbon. And so when we take that into consideration, and Anderson in 1995 did that, he created a best cell that's broken up by these proportions down below. You will note, if you actually took the time to calculate that, it does have a little bit over 100. He's using that estimation basics, but it is slightly above 100. But when we do this, we actually get that there's 114 to 106. So the value for him, the respiration quotient, is 1.07. That 0.07 is 7% more oxygen is being consumed and utilized per amount of carbon. So it's actually slightly impacting, right? A lot of people don't think that would be important, but we're going to show very soon how just that little bit more has strong implications for the global ocean when we consider it. So this is what we have for the respiration quotient. That's great. It took about a year to figure out how exactly to measure this in a sample. Part of the problem is we're dealing with the ocean, and the assay, in order to get the particulate chemical oxygen demand, doesn't like salt. That's problematic. However, we were able to kind of figure out a way around that. And what we're doing now is we are filtering seawater onto GFF filters, so glass fiber filters. And the idea is that any particulates that are stuck onto that filter then are going to be placed into a tube, and we allow for the deoxygenation process to occur. We can then measure how much oxygen was utilized at the end, very simply. And we divide that by the particulate organic carbon. Now, this is a value we get all of the time in the ocean and in soils using an elemental analyzer. And we divide the two values, and we now have the respiration quotient. Great. So what are the first measurements? What do they look like? So we went to the Eastern Pacific Ocean. So we started in Baja. So the cruise transect was P18, Baja, California, down to sea ice, came back up to Easter Island. And what we see here is that on average, the respiration quotient is actually 1.15. So that's a little bit more than that Anderson estimated value of 1.07. So in fact, the average in the ocean, in the Pacific Ocean, is slightly higher. However, it does have a very good range. So we can get as low of a value of 0.73 and as high as 1.54. We also see that we are getting a linear relationship with temperature. So as we increased temperature or moved towards the equator, we found that values were higher in those regions compared to in the Southern Ocean. That in and of itself was pretty amazing, because we had no idea if any environmental factor would matter at all, or if this value would even vary. Most data says it wouldn't, and we found that it did, and that we have this pretty decent relationship. Now, I say decent, and yes, we're looking at this, and some people might say that's not that great, but we're looking at about a point, an R square of about 0.2, which is pretty good for ocean data. Okay. So from this, we were able to actually generate some hypotheses, because of course, temperature wasn't the only thing that we took. We took nutrients, and we took nutrients being both phosphorus and nitrate. And so what we can actually do is create some hypotheses based around this idea. This on the side here, this cartoon is a little bit of a spoiler, just so we can walk through it very simply. The first hypothesis is that temperature has this effect, right? So we saw that in the Pacific Ocean. We see this increasing trend. So maybe on average, we know that the respiration quotient is going to increase with increasing temperature. The second thing that we know is that a lot of regions in the ocean that exhibit high temperature, like the tropics, are also going to exhibit low nutrients. So if we see an increase in nutrient limitation, lower amounts of nutrients, then we're going to see higher respiration quotient. However, that's very broad, right? Nutrients are very specific. We have phosphorus, we have nitrogen, we have iron. And so when we consider nitrogen, for example, from biofuel research, we know that as you decrease the amount of nitrogen available, a lot of phytoplankton will increase their lipid content. And by doing that, they're going to potentially increase their respiration quotient, because lipids have the highest respiration quotient per biochemical component, so compared to proteins and carbohydrates. And so we can create a schematic that looks like this. So we can partition out this very simple version into, as we increase nitrogen limitation, respiration quotient is going to increase. However, when it comes to phosphorus limitation, we're not really sure. Data can show different things, because phytoplankton like to act very differently when it comes to phosphorus. And so we're not 100% sure, and I like to put just a question mark, because who knows? We'll see what we find. The third, and probably one of the most funnest things, is cell size. At the end of the day, what we're measuring is particulate organic matter. We are looking at phytoplankton. All of our samples are taken at the surface. And so by doing this, we need to consider, well, does size matter? Of course it does. When we look here at an allometric relationship between the abundance of macromolecules and that of cell volume, there's actually a very interesting process, or a very interesting pattern that we see. The carbohydrates and proteins will increase very in a paralleled way. However, lipids will not. So what we find is that if you are a cell that is in small volume or a high surface area, you tend to have more lipids. Now here I've actually written out the different values for the respiration quotient for each of these biochemical components. And with that, we see that lipids, again, are the strongest. So when you have high amounts of lipid content in such a way here, you're going to have a high respiration quotient. And that's similar to what's seen in cyanobacteria. And comparatively, the opposite is true in a diatom that has a lower surface area to volume ratio and therefore a lower respiration quotient. So we're able to generate all three of these base hypotheses just from measuring in the Pacific Ocean. Now we're going to go ahead and test to see, well, can we validate any of these? What's most impactful in the Atlantic Ocean, or is it exactly the same in the Pacific? So what do we see? We're going to specifically look at the average respiration quotient, and we're going to go ahead and compare that to the Pacific Ocean, because we want to know whether each region, so the ocean basin itself, is different. And then we also want to test our hypotheses to see which one best explains it. Here we're looking at the AMT 28 cruise transect. This went from Plymouth down to the Falkland Islands. This has 715 stations. These samples are taken hourly. A ton of work went into this. We have single particulate chemical oxygen demand values for all stations, except at noon where we took triplicates. We were trying to test diel cycling, and it didn't work as well as we thought it would, but that's okay. Everything else is done exactly like it was in the Pacific Ocean. And for anyone who's never seen a cruise ship, they are massive and super cool. This is what they look like. And a ton of research gets done on them. So on average, I love that we got basically the same value. So we get an average value of 1.16. So that's not statistically different than 1.15. However, here we do see that the spread has increased quite extensively. So we have in the light gray, so this larger space that goes all the way closer to 1.9, we actually have the Atlantic Ocean, and in the darker color in the front is the Pacific Ocean. Now this could be because we're taking hourly samples and a mass amount more, but there's also a little bit of a spoiler, there's interesting factors that can cause such high values, and we are going to see what those look like in a second. So we're going to go ahead and test, well, what are our hypotheses, right? We have these three still. Temperature. It was so disappointing. Yes, we can fit a line along this data, and it works. It's not great, not very much at all, but we do see that on like the intercept and the slope are relatively decent when we compare that to the Pacific Ocean. So how temperature is acting on the respiration quotient is similar. So that is not in question. Temperature does have an effect on respiration quotient in a specific way. Not necessarily sure yet what exactly that is, but we do know that it exists. However, when we consider the nutrients, this is where things get really interesting. Now, in the UCIC 2021 paper, these are data that are taken at the exact same time as our cruise transects. And this is looking at a mass amount of cruises. To make my life easy, I just put them all up there. We're looking at limitation, specifically categorized from when Prochlorococcus is going to show nitrogen, phosphorus, iron, co-limitations occurring. And so each of those is a different color. So we have phosphorus is in blue. We have nitrogen in this yellow color. Pink is iron stress. And so what we can look at when we take into consideration our specific cruises, which I pulled out specifics, we actually see a lot of that yellow and pink. So we have a lot of iron stress and nitrogen stress in the Pacific Ocean. Whereas when we go along the Atlantic Ocean cruise transect, we now have a lot of phosphorus stress in addition to that iron or nitrogen stress, and a few spots of iron stress, but not too many. Okay, so we're going to use these indices in order to determine if there is an effect. Now this plot is a little bit of chaos, and I do apologize, but it is one of the best ways to show what is happening. So let me orient you first. We have the Atlantic Ocean north going down to the south on our X, and we have a normalized parameter on our Y. The blue coloring, which is up on the above, is temperature. The index for phosphorus stress is in red, and it is the solid line. So if you are colorblind, look at that solid line. And the nitrogen stress is in the dashed line or in green here. So what we're able to see, oh, and then of course we have the respiration quotient is the smooth black line that's very prominent because that's what we care about. When we have nitrogen stress, so when the green dashed line is higher than that of the red line, which is very low in this area that I've picked, we see that the respiration quotient is actually above temperature. Now, what we do see at the beginning and end of the cruises, when there's really no limitation happening, is it's tightly coupled to that temperature line. So we see that there's this extension above what we would expect if it were to follow temperature alone. Interestingly, when we have very little co-limitation to very little, like we're fighting for which one is actually more limiting, we see very consistent respiration quotient to very little changes. However, and I think this one's really interesting, and we were not expecting this whatsoever, is phosphorus. So when we have phosphorus limitation occurring, so here we have this red line going very high compared to this decreasing in the dashed green line, we see that the respiration quotient dramatically decreases. Now, why might that be? Most likely it has to do with the way that phytoplankton are able in their biochemical components. They're shifting gears and going away from lipids, right? They're not able to get those bilayers built because of phosphorus, and they're putting their stores either storing other biochemical stuff, or they're actually creating proteins and enzymes and things and actively working. And so this might be one of the reasons why we see this change in the respiration quotient, specifically for phosphorus limitation. Okay, so this means that it really matters. We really need to understand which specific nutrient is limited in the region we're looking at or in the sample we're looking at. And this, of course, can hold true. We expect, I should say, that this will hold true in culture work as well, though that is future work for me. But we do believe that that will hold true. Now, although this tells us a lot, right? So I was telling you we have very high respiration quotient, why potentially nitrogen limitation, but we cannot forget the cell size because we care about who is present. Now, the first thing that I want to do when orienting on this is to discuss what we're seeing. So we do, of course, have latitude again. We have the ratio between the small and large phytoplankton. So at the time of sampling, there were samples taken for the cell sorter, and we were able to break down, there are these large phytoplankton, these small phytoplankton, categorize them, give an estimation of how much carbon is in those samples, and then create a ratio around that. Now, one thing that you would see when you look at the purple line here, and that is where we have data as well as smoothing going on, is that it is pretty much the same when we look at both the southern and northern hemisphere. And that's something we expect, right? We expect from like a biogeography point of view, that they're going to be relatively similar as far as size classes, give or take. When we look at ratios, they're going to be pretty much the same when we look around the equator. However, what we see is that, again, if we expect there to be more large phytoplankton, so a ratio that's lower, right? So we have less small phytoplankton to large phytoplankton, then maybe the respiration quotient is going to decrease because we have more large phytoplankton. The interesting part is this is also the region where however, we expect to see that this would not necessarily follow that, because here we have the exact same ratio, right? So this ratio and this ratio, give or take, are about the same, and yet the respiration quotient is completely different. The reason that is here we're looking at spaces that are phosphorus limited, and therefore we're seeing that the physiology is actually what we're measuring somehow. So we're getting physiology responses, not just who's there, it's how they're acting. Okay, and so it's important to consider that, because now we see that we have this hierarchy. It's extremely complicated to decide which is our best explanation at its core. And if we had to only put one thing into a model, it would be an equation that includes temperature. if we're allowed to get crazy and computational powers were amazing, we would add in this idea of nutrient limitation, and we would add in cell size. Because remember, temperature is telling us who can live within this region. The nutrient limitation is going to be like, how are they acting? How are they doing? What are they able physiologically to do? But then the cell size is going to give us constraints. So we can see specific constraints on processes based on their cell size. Okay. Now let's talk about the future a little bit, because this is all well and great, but we all love future projections, especially if you do any climatic work. You got to say what happens when temperature increases, because why not? So what we have here is a community Earth system model. And this model contains a physical climate, which has atmosphere, ocean, land, sea ice. We have specifics within a lot of chemistry and atmospheric processes. There is ecosystem aspects. There's biogeochemical processes also built into these models. Now here, normally the respiration quotient is set to 1, because that is what Redfield found, and we in oceanography love to follow Redfield. So what we decided to do was use the exact model as is, and instead we got the respiration quotient and we adjusted it. So we went from a value of 0.8, so this is one run scenario, 0.9, another run, of course the 1 value, etc. And what we find is that the global inventory of oxygen in black is decreasing as you increase the amount of oxygen. It seems obvious. As you increase oxygen demand, you're going to decrease the amount of oxygen that is available because they're utilizing it. And so we expect to see that that would happen. And of course, we also then expect that the oxygen minimum zones are going to increase. And so we're going to see larger amounts of these oxygen minimum zones based just simply because we're adjusting this value. And what we see is when we have this current value, because now we have a current value, right? So we're moving essentially from this one line, which would be here, and our actual estimation is 1.16. So this green space, if we were to include that temperature is going to increase by 2 degrees, which that at this point is a little give or take, and the fact that we know that nitrogen limitation is going to potentially increase in the future, we can now estimate that the value is closer to about 1.28, give or take. So moving towards this purple line. So we see this normal scenario as of today. If we were to move it to the future scenario, we actually would see a pretty dramatic decrease of 30% of oxygen globally. So that's just based off of right now what we're saying our models would look like. Here we have a map showing the changes in the oxygen. So any values, of course, that are blue are a decrease. There's very, very small areas that show red. They do actually pop up there, not very many. And where we see oxygen decreasing the most, it, of course, is the equatorial Pacific. We also see the Bay of Bengal and the Arabian Sea. Now, for those who don't do oxygen or ocean research, these areas already see large spaces of oxygen minimum zones and changes in oxygen constantly fluctuating. Now, because that exists, we're going to see this additional potential influence. Now, I am being a little doomsday in the idea that I am saying, oh, it's 30%, but I, just as all modelers did, was changed the value, right? Statically changed it. So the changes we may be seeing actually could be a little bit less dramatic as this. However, this does take into consideration that we are already changing from a system that is set to 1 and moving to 1.16 anyway. So we're already overestimating how much oxygen is there. So keep that in mind. Why does 30% matter? Okay. At the beginning of the talk, I briefly said that this is super important to marine life. We're going to see all of these things happening, right? They are all true. If we see lower oxygen, and we don't need to wait for a future scenario, right now we know that that's happening. We're going to see that coastlines are going to have major issues, ours in particular. For anyone who likes going to the beach, it's going to be very interesting in the next couple of years because there's not going to be as much biological productivity and diversity. We're not going to be able to see some organisms just hanging out like we used to. I went to school in Monterey Bay, and you could very easily go whale watching during during whale season. Nowadays, it's very hard. You're very unlikely to see them. That's partially due to the idea that we have changing temperatures and changing oxygen values. And of course, both of these are going to have very strong implications on our fisheries. Now, to kind of go a little deeper into fisheries, because I think that really is something we need to dive into and understand. Here you're looking at current historical and projections. So we have historical in gray, projections for a climatic scenario that is best case. We ain't gonna hit that, I don't care what anyone says. In blue, and then in red, we see worst case scenarios. Will probably be somewhere in the in-between. The first plot specifically, and this is in time. And what we have here is the change in biomass in fish. Okay, so fish biomass changing. And what we're seeing is that there, in about 5 to 7 different models, we see that there's not very much consensus, first of all. The darkest color is the average, and then we have the error in the different model types in the kind of, I don't know, lighter coloring in the background, the shadow color. And here we have a lot of overlap between the two. Now, this is just a basic projection that a lot of fisheries do. In most recent years, though, however, they've been able to do a little bit more constraining on those models. And so what they've done thus far is they've not only taken into consideration more processes that before they hadn't had in the model, they're also able to expand. So there's more models coming out with different processing. And so we see that there's actually a very distinct possibility that if we stay in this kind of best case scenario, the amount of fishing, the fisheries themselves are going to lose less biomass over time compared to a worst case scenario where we see these dramatic decreases in the biomass. Okay, why do I say that? Why does this matter? Well, these models do not contain oxygen currently in any way that's constrained in these models. And if they do, they're very complicated. And they're not something that looks like our respiration quotient. So because they're not taking into consideration that the oxygen is going to be adjusting, some of these fish counts or biomass counts are not necessarily as realistic as we want them to. However, this is still research that's being very much done at the time. Like, there's a lot of people in the fishery world doing these models, trying to get the best estimates. Fish are complicated. I do not touch fish. Well, I can touch fish, but I don't touch models and fish. They can do their own thing. They're really cool. But in addition to the fishing, we also, again, need to think from a recreational point of view, right? We do like to go to the ocean. And this is a picture of Monterey, and we have dolphins and birds. And we're probably going to expect to see something a little bit more like this on the gloomy days, but also showing that less diversity is going to be seen. And again, it's very, very common, very possible, given where we're looking at along our coastlines. This is all great. I'm going to toot my own horn and I say this is all great. But I was a little unsatisfied with some of our cell data, right? I wanted to know, if we're looking at phytoplankton, this is at the fundamental of what we're measuring, why is it that we can't really see any differences when we're looking at the different regions? And so what I decided to do is work in conjunction with Mike Lomas at Bigelow Marine Station. And we grew 4 different types, species of phytoplankton, specifically looking at a cyanobacteria with a flagella, a circular diatom, and a cubic diatom. And what we actually find is the more complicated these cells get, their respiration quotient decreases. That was very surprising because we don't really understand why that would be true. Now, these are all grown at their optimal temperatures, and they're all happy when it comes to nutrients. However, we're seeing that a cyanobacteria that's grown normally, another cyanobacteria with a flagella, that flagella, of course, does contain those different biochemical component structures. And because of that, we now see that the averaging of that becomes a lot lower. The other thing we found, which I think is also really great, is when we looked at this against its optimal temperatures, we actually found that if you grow phytoplankton at their optimal temperature, those increases are going to result in increased respiration quotients. And so that may mean that fundamentally, if you're found in a specific region, you may have a temperature constraint on what your respiration quotient can be. Now, this is very fresh off the presses. This is data that we're just now playing with, but we are seeing that there may be these more physiological responses that we need to pay attention to. And so this is some of the work that I'll be continuing in the near future, and then hopefully later in my lab, we'll be able to do a lot more on this work. So in summary, we've been able to create a very reliable method to quantify the respiration quotient. It will work in all spaces within seawater. We also have a high hypothesis that it's going to do great in aquatics. So we do want to test that again in the future. We have not yet done that, though we have tested this in the Indian Ocean and on the coastline, and it does perfectly well on both of those spaces. The respiration quotient on average is approximately 1.16. Now this needs to get put into models. I know the modeling community who think, okay, we need to put this in there, yes, but it's not as a high priority. Hopefully with some of our papers, we'll be able to make them put it in there because it's extremely important. The other thing is there is basin variation that does exist. And so we need to consider how we would act when that comes into consideration, right? Do we put certain regions at their average value versus another basin, give it its average value, or can we give a single value across the board? There is this hierarchy that does exist in the environmental conditions and how it's going to be controlling respiration quotient. Again, something that we need to consider every time we're doing sampling, that we need to get all of these factors to really understand the process and what's happening. Of course, this has very strong implications on both the ecosystem health, not just from the oxygen point of view, but also from a nitrogen, phosphorus, fish point of view. Everything seems to matter when we consider how this value is going to change. And so with that, I'm going to say thank you for your attention. I will say that I do have my email. So if you are on Zoom or here and you don't get to ask a question, please feel free to email me. And I do have two papers. The AGU Advances is the most recent that just came out last month and has to do with a lot of the work I talked about today. So with that, again, thank you for your attention, and I'll take any questions. Yes.
I just wanted to make sure I understood the process of the base. So basically, you don't have enough phosphate to make the bilayers, and this has then an indicator for cell size. And you want to make bigger cells then?
Yeah, so that would be one that's one hypothesis. The other is that the cell could potentially be storing something else in its vacuoles, and that would give us something very similar of a result, right? And so it could be that it's either that it's just not growing in small, it's getting big, or it's the vacuole itself is getting, because of the storage component, is becoming larger. Yes.
Are there any differences in the phylogenetic distribution of phytoplankton by latitude?
There are. I think it depends on what specifically you're looking at. So one of the best examples I can give is Pokerococcus has very clear distinctions between being high light, low light. They have ecotypes based on both high light, low light. It dives in deeper, whether they can be in low amounts of iron, small amounts of iron. And so we've seen that very clearly in Pokerococcus. A couple of other species, it is, I shouldn't say, there's not ecotypes, that's not as well defined, but phylogenetically, there are differences. We would expect that some of those differences would cause there to be changes in the respiration quotient. We don't know yet, mainly because, and I admit this completely, a lot of this work, this is the first time we've been able to get this value. And so we haven't gotten that far yet. and the field is such at its baby infancy stages. But it would be really great to see if we could grow different types of Prochlorococcus because they're so well used, and see if those ecotypes actually are changing. Yeah, it's a great question. Thank you. Go for it.
So in what you've done, really have yet to address the distinction between what the deep water systems in the middle of the areas relative to shallow. Is that correct?
I'm gonna say yes, because the best way to do that would be a transect that's going more along a lateral, right? And so if we can get, instead of being, yeah, we all of our transects are definitely done up and down. If we can get across a space, that would be the easiest way to do it. I think I've thought about it before, but yeah,
But the data you're getting are from constant depth or
they're all surface based. Yeah, so relatively constant. It would also be really cool to do it in the water column first, just in any space, but then also, of course, to go through an oxygen minimum zone. That would be amazing, because then we could see if there's actual differences when you have low oxygen in the space anyway. Yeah.
That's really interesting. You look a lot at how variation in the phytoplankton biomass affects the value of the method for studying how much oxygen is used to inspire the biomass that's there. I was wondering what the effects might be on changes in the bacterial community, on what that ratio actually turned out to be. Are different groups of bacteria more or less efficient in how much oxygen they're using to consume that biomass?
Yeah, that is a fantastic question. I feel like I'm going to tack it to my list of proposals, because that is definitely one of those questions that would really tell us in deeper water column how much of this process is actually going to what is traditional deoxygenation and how much is not. So presumably, from my very little knowledge of bacteria, they are different. They do have different efficiencies. And so they are going to be a little bit messy in some cases, not others. They can be a little picky, I think, when it comes to particle sizes. And so that also would change, right? If a particle is coming down and it's already been grazed multiple times, it has less goods. And so the bacteria are less likely to get them. There's also this efficiency anyway, with at some level, they may not get to this first layer of bacteria. There's a second layer of bacteria that will then continue to consume anything that goes past it. So there may be a difference between those as well, because there are very different types of bacteria living at those levels. So yeah, that's a really interesting question. Yeah.
oh, there might be a downside.
No such thing.
But okay, now you
No such thing.
on the focusing
Yes.
zooming out and but for finding context.
Yep. Yep.
So these focus same that was part of focus that we the same as the respiration focus.
Like how much oxygen is being produced per carbon?
Yeah.
So I feel like the answer is it's not the same. Like the value isn't going to be the same as the respiration quotient, mainly because the efficiency of producing oxygen is going to change with time for the phytoplankton themselves. And then different types of phytoplankton are producing more or less oxygen depending on what they're doing, the environments they're in, and if they're stressed. So in that sense, not exactly, but it's more than likely impossible to do something very similar to figure out what that variation would look like. I think some people know the answer, depending on their specific species, how much oxygen they can produce, but I don't think there's a collective answer yet. Yeah, good question.
You didn't mention refractory or export. Is there, do you want to say anything about that? Or something?
I think the answer is more, we're not sure is part of the problem with both of those. Because we know that, so I've hypothesized before that if we continue to see, like, we'll use export, for example, you have export coming through the water column. Of course, we don't know what the respiration quotient is going to do as it's being exported. However, if presumably the respiration quotient would decrease because it's going through the water column, then it's possible that as that process is happening, we may be actually using a lot of oxygen on the top and a lot less, and basically creating these profiles of respiration quotient utilization of oxygen. Refractory, not sure. We did have we had a discussion in the newer paper. We have a discussion talking about the differences between looking at like DOM, dissolved organic matter, and how that potentially plays a factor, because our samples technically can be overloaded where there is some amount of DOM on the filter itself. We also don't necessarily do a blanking technique. So for those who are more familiar with that, a lot of times when we do particulate organic carbon, you want to either dip your filter into the water and just what is the baseline carbon that's present. We don't do that. However, when we originally made the assay, we took into consideration what the estimated value should be, and it's extremely tiny. And so there's probably not as much just leftover carbon on the filter by itself, but DOM could have an effect. We're just not quite sure yet what that is. Yeah, great question. Yeah.
Open up space if there are questions from students or postdocs in the room for Allie to hear about with research or charts.
Yeah, whatever. Yeah. And there really is no dumb question, because you'll probably stump me, let's be real. I don't think we did. I don't remember there being any. Easter Island, we saw penguins. Those are cool. Easter Island is really cool too. Yeah. I don't remember there being. They're also not the easiest thing to see. One, the cruises are going. Two, pretty far down, and the shape of sharks in the water column is going to be different. Yeah, but. Are there any questions on the chat? I'm gonna just pop it. Nope. Okay, cool. Just checking.
So further questions, I guess we'll wrap up. Please,
Awesome.
thank you, Allie. There will be no seminar next week, but we're back again in 2 weeks, right?